Hello, can we start now? Hello, are you there? Hello, are you there?
Yes, ma'am. I am here always.
Okay, and you can see the slides. I think it now it
Yes,
is.
yes. Slide is visible.
Okay, so sorry. Today we can discuss the MCQ questions first, and then we can discuss one paper. For this week, I think Sir has given 3-4 reading assignment, but only one paper we can cover today. In the other papers, if you have any doubts, you can ask that we can discuss in today's session itself. And the paper also I have put in the chat box what we will be discussing today. First question, pick the odd one out.
Ma'am, please move to the second slide, question slide. Then I can see yours.
It is which slide it is in? It's not in the question slide. For me, it is showing in question slide.
Yeah, ma'am.
For which slide it is showing for you? For
The
me, it
first
is
slide.
showing question slide only.
First slide.
Okay. Now can you see? But now it is not in.
Yes, ma'am.
Okay, now I'll just go to full screen mode. Now you are able to see the question slide.
Yes, ma'am.
Okay, great. You can type the answer or you can also just unmute and say it. Yeah, you are correct. The answer to the first question is Emerin. So emerin is a mechanotransduction protein, but it is present in the nucleus. The other three, ARC23, filamin, and actin, these are all present in the focal adhesion. So the odd one out is emerin because others are all focal adhesion proteins. Let's go to the next question.
Ma'am, please change the slide.
Can you repeat?
Yeah, please change the slide.
I have changed it. It is in second question now.
No, I'm not. It's not showing to me.
In slideshow mode, I think some problem is there. I'll go to slideshow again.
Yes.
Now
Okay,
it's
okay.
working.
Just a minute.
Okay. Okay. Yeah, second question answer is correct. So when it comes to migration, there are like three main processes. Um, Polymerization, that triangle mode that sir was saying, protrusion, addition, and contraction. And in this case, it is a non-adhesive substrate. So addition won't be there, and contractility also won't be there, because only if it can adhere to the substrate, it will be able to generate the contractile forces. So if it is a non-adhesive substrate, then the main method will be polymerization. So the answer is option A, increased actin polymerization. Third question slide is visible. You can see the third question, right?
It's loading.
Okay. You please type the answer for third question, then we can discuss it.
Ma'am, but the question is not showing, it's loading. Actually,
Maybe
it's
it's not coming.
maybe some network issue from your side. It's loading.
Okay, I'll just copy paste in the chat box, then you can see it meanwhile.
Yes, now it's showing. Just a minute. No, don't need to copy paste. Now I am see this.
Okay,
TBC.
okay. I think in slideshow mode it is not coming properly. So I'll just keep it without slideshow mode. Yeah, sorry. Third question is also correct. So in the 2D form for amoeboidal migration, all of it is possible. So everything will be, it will be combination of all three: protrusion, adhesion, and contractility. So the correct answer is option C. Let's go to the fourth question. Yeah, fourth question is also correct. These are the focal adhesion proteins and they are all connected to each other. But whether this connection between integrin and talin and actomyosin filaments, it depends on the substrate stiffness. So in the higher substrate stiffness, the connection is more prominent and we see a catch bond phenomenon. But in the lower substrate and less traction forces, it is not connected. So there is a threshold. Only above the threshold, the integrin, talin, and actomyosin will be connected. So it is not. It is only a transient connection. So the option correct answer is option B. Let's go to the next question. Fifth question, they are asking about cell adhesion forces in 2D and 3D. In 2D and 3D, the main difference is that in 2D, the cell is having just one plane to attach to, like because the extracellular matrix will be only present in the bottom. In the top, nothing will be present. But in 3D, the ECM is present in all the sides of the cell, so it can make attachments in all 360 degrees in all directions it can attach. So because in 2D, in only one plane, only one part, the cell can make the addition and attach to the substrate. In 2D, the forces will be higher compared to 3D because it needs a support so that it can get the nutrients and everything. So it has to make a very strong addition and only one it is having only one possibility in only one plane. So the addition forces will be stronger in 2D. Okay.
Okay, because it has only one plane, so adhesion forces is on strong backbone.
Yeah,
Strong
exactly.
backbone.
Yeah. Let's go to sixth question. Question 6, you are correct. For wound healing, you can do the wound healing assay or the microfabrication. So, I can just draw and show quickly. Draw option. Okay, I think it is not having draw. Okay, I'll just explain like that. So scratch assay, wound healing assay means it is basically a scratch assay where you will be having a monolayer of cells, and using a pipette tip you will just make a scratch in the dish. So the cells which are touching the pipette, they will be removed and there will be a gap, and the cells will move to close the gap. So that is option A, wound healing assay. And microfabrication means it is based on PDMS. So PDMS is like an elastic substance, and so what they will do is they will just if you are growing cells on a glass bottom dish, they will just put the PDMS. It is like a sticker. So one side they will put the PDMS. So one side will be covered, and then after putting the PDMS, they will add cells to the glass bottom dish. So when you do that, only the side which is not having the PDMS, the cells will attach. The other side with PDMS, the cells won't attach. And then when they remove the PDMS, the cells will go to the gap where the cells are not attached. So microfabrication, it is based on these PDMS stencils. So it is like some kind of a sticker, and when you remove it, the cells will sense a free gap and they will go there.
Can you please repeat once more about those things, wound healing assay and microfabrication in little bit?
Okay, actually, do you know if there is draw option in this one Google slide? I'm not able to find. Then I can draw and show. That will be easier. Okay, it is not there. So first method is scratch wound assay. So first, in scratch wound assay, you will grow the cells normally, like using a glass bottom dish. And once the cells have fully grown, using a pipette tip, like you can use 200 microliter tip or 1 ml tip, just make a scratch. So when you make the scratch, wherever the pipette tip is touching the cells, those cells will be removed. So there will be a gap in the monolayer, and the cells will, like both the sides, the cells will try to come and close the gap. I will just put an image here, then it will be easier to understand. Yeah, so something like this, like that circle that they are showing here, that is the glass bottom dish, where first you will culture the cells normally. And once they have formed this monolayer meaning the cells will be touching each other, there should be no gap. So then we will say that it is confluent. And once they have become confluent, using a pipette tip, you will just draw a line, or then it will make a scratch, and the cells will slowly come inside to move to close that gap. So this is the first method. It is the wound healing assay. And the second method is using PDMS. So PDMS is like, it is something like plastic basically. You can make different shapes and sizes of PDMS. So what they will do is before seeding the cells. So here they have seeded the cells everywhere in the glass bottom dish. But if there is PDMS, suppose Suppose this is the glass bottom dish. Before seeding the cells, you will make a PDMS shape which is covering half of the dish. Or depending on the experiment, you can change the shape of the PDMS. So you will just stick this PDMS on the glass bottom dish. And then when you seed the cells will only attach in this part wherever there is no PDMS. So on the right side here, the cells won't attach. And on the left side, wherever the PDMS is not there, only in that semicircle the PDMS will attach. So first we will seed the cells, and then like this it will in figure A it will become confluent. But the difference will be that only in one half circle the cells will be confluent. In the other half circle where the PDMS is there, the cells won't be confluent. And then once they have reached confluency, you will remove the PDMS. So once the PDMS has been removed, so only one side the cells are there, left side only cells are there, right side the cells are not there. So the cells will go from this side, left side to right side. So this is another method for studying collective cell migration. They will try to close the gap. So that is the microfabrication method, option 2. Now is it clear?
Okay, so microfabrication method is carried out by the PDMS.
Yes, yes.
And wound healing normal only for the normal cells.
Yes.
Okay. Now it's clear.
Okay. This is actually 6th question. I've mentioned already. 7th question is. Yeah, now let's go to the 7th question. So this question is about Yamanaka factors. So these factors are like if you add them to stem cells, then depending on which factors you are adding, you can change the fate of the stem cell. So these are called as Yamanaka factors because he is the person who has found out about these factors. So you just have to add them to the media. And depending on which of them you are adding, they can become a mesoderm cell like fibroblast, or they can become an ectoderm cell. And these factors are these 4 chemicals. So you can just add them to the media. So, and these will dictate your stem cell fate, whether it is going to become fibroblast cell or neural cell or bone cell, etc. Others, option D, FBS, CGF. FBS is this fetal bovine serum. So these factors we add to cell culture routinely. It is not just added for stem cells. But option A, these factors, these if they are added specifically for stem cell differentiation. So the answer is option A. These are the transcription factors actually, which is controlling which genes will be present, and some genes are needed for like different cell types will be needing different types of genes. So all these are actually the transcription factors. And in our body also these transcription factors, they are on in some cells, but in some cells they are off. So so apart from doing it in the body, we can also do it in cell culture by giving them separately. That is the concept behind Yamanaka factors. Let's go to the 8th question. So this collective cell migration, this question is about collective cell migration, which we were discussing in the previous question. Like for example, during wound healing and all, the cells have to move as a group because in the epithelial tissues inside the body, they are not present as single cells, but it is a tissue, right? So they are all connected with each other. And in collective cell migration, the special property is that the cells will all move together as a group. They won't, it is not like one cell will go right side, one cell will go left side, but they'll all coordinate with each other. And as a group, they will close the wound. So how it is happening is that so there is a one concept called leader cell and follower cell in collective cell migration. So whenever there is a wound, some cells will become like leader cells. So these cells are, they are bigger in size and they can exert higher forces. So basically it is like these cells will become leader cells and they will pull the cells behind them. And the cells which are behind the leader cells which are getting pulled, they will follow the path of the leader cell. So they are called as follower cell. And the main one of the main characteristic of the leader cell is that it will exert a high traction force and high cell force to pull the cell behind them. So the focal adhesion, so for exerting forces, the cells will use the focal adhesions, right? So the leader cells will always have better or more prominent focal adhesions than follower cells. So option C is correct. And option A is also correct. In collective cell migration, leader cells are more mesenchymal. Mesenchymal meaning that they will follow the mesenchymal mode of migration. So in the lecture, if you have watched the videos, there are like two main kinds of migration: mesenchymal migration and amoeboidal migration. So mesenchymal migration is the migration where first there are these lamellipodial protrusions, and then there is, it gets addition to the substrate, and then there is a contractility in the behind of the cell, and the rear end of the cell detaches from the substrate, and therefore the cell moves forward. So if you just look at the leader cell during collective cell migration, they will also follow this mesenchymal migration. Even in the leader cell, you'll be able to see the lamellipodial protrusions and adhesion forces and contractility. So option A is also correct. And option B is not correct because, like I was telling before itself, during collective cell migration, they will migrate as a group. So the cell interactions, it will still be together, but they will still move together because they are moving together. The cell bonds, the bonds will not be broken. And option D is not correct because leader cells, like when the wound has been formed, at that time the leader cell may be like some 2 to 3 cells inside, and then from the inside they will go to the outside and then they will become the leader cell. So the correct answer to this is option D. Sorry, option D is wrong. Yeah, the correct answer to this is option A. Is it clear?
Yes, it is correct, but side by side option 3 is also partly correct, right?
No, option 3 is not correct because in the leader cell the focal adhesions will be more prominent. I was telling, no, the leader cells will exert high forces to pull the cells that are behind them. So because they are exerting high forces, to exert forces you need the focal adhesions. So that is why the focal adhesion will be more prominent and bigger in the leader cells than follower cells. But option 3 is telling the opposite. So 3 is wrong.
Okay. Leaders.
Okay, let's go to
Okay,
8th
ma'am.
question. Okay.
It should be question number 9.
So this question is about stem cells. There are two kinds of stem cells, adult stem cells and embryonic stem cells. Embryonic stem cells means during the developmental stages of the embryo. So the initial cells that are present, they are called stem cells because they are the ones which are differentiating to give rise to all the organs. And these have the special property, like I was just saying, that they can give rise to any of the organs. So this property is called pluripotent because they can give rise to all the cells of the endoderm tissues, ectoderm tissue, mesoderm tissue, everything. So this special property is called pluripotent. But the other kind of stem cell is called adult stem cells. So these are present in the tissues of the adult body, but they are present very less in number. So, for example, if you look at the intestine, the cells of the intestine are they are getting constantly removed because they are the intestinal lining. It is always in touch with the food that is passing through. So, to replace the cells that are being removed, there are stem cells in the the villi, right? In the bottom of the villi, there are stem cells. So, as the cells in the top are getting removed, the cells in the bottom they keep differentiating. So this is just one example in the intestine. Similarly, in all the organs, there will be some fraction of stem cells, although that fraction is very less, so that in case there is an injury, these cells can differentiate to give rise to the cells of the tissue. So these are called adult stem cells. But the limitation of adult stem cells compared to embryonic stem cells is that the adult stem cells can give rise to only certain cell types. For example, if you take the intestinal stem cell, then they can only give rise to epithelial tissue. They can't give rise to bone tissue and all. So these cells, they are the adult stem cells are called as multipotent because only some cell types it can give rise to. And so stem cells are very promising for regenerative medicine, right? Because they can differentiate into any cell type, especially the embryonic stem cells. But the problem with the embryonic stem cells is that you have to remove it from an embryo, right? So that may damage the embryo and the embryo may not grow properly. So with the embryonic stem cells, there are many ethical concerns but adult stem cells, the concerns are less. So if you, that is why adult stem cells are more ethical to use, because this concerns that you have with the embryo, that the embryo may not be healthy if you remove the stem cells, that is not there in the adult stem cells. And option 2 is not correct, because adult stem cells are multipotent and embryonic stem cells are pluripotent. Option 4 is the correct one. Embryonic stem cells can give rise to ectoderm, endoderm, and mesoderm. Let's go to the 10th question then. So 10th question is also about sorry, stem cells and differentiation. So HMSC means it is a type of stem cell. It is a type of adult stem cell actually. So one major question in biology, especially in the stem cell field, is that how can you determine which what to which cell type the stem cell is going to differentiate. So this is based on many parameters, like many factors. For example, which factors you are giving in the cell culture, and what is the cell density when you are seeding it. Because and it also depends on the substrate stiffness. For example, this HMSCs, people have shown that in stiffer substrates, like for example, like 100 kilopascal gels, they become bone cells or osteogenic lineage. Because if you look at the body, in our body, the bone tissue is much more stiffer. So if you make the cells to grow in an external environment which is mimicking the bone stiffness, then the stem cells, they are more likely to become like bone cells. So they are able to sense the environment and understand that we are in a stiff environment, so we should become a bone cell, something like that. And similarly, if you plate them in a softer environment, like 4 kilopascal or 1 kilopascal polyacrylamide gel, then they will become more like fat cells or adipogenic cells. So earlier, the people were thinking that stem cell differentiation, it only depends on the chemical signals. But later in mechanobiology, people have shown that it depends on the substrate stiffness as well. So stem cell differentiation depends on both the soluble factors or the chemical factors that are present, as well as the substrate stiffness. And option 4 is correct. Stiff bone is a very stiff tissue and it is having a stiffness around 100 kilopascal compared to the brain, which is having like 1 kilopascal. It is very soft. And option A is not correct because in soft substrate it will differentiate into adipogenic, and in stiff substrate it will differentiate into osteogenic. So 10th question, the correct answer is option D. One more extra question is there that you can see whenever you are taking the slides. But now we can go to the reading assignment. So till 7:40 p.m. you can go through the paper. Then from 7:40 onwards we can discuss it. I'll just put the link also. The link is yeah. So 7:40 p.m. we can start to discuss the paper.
Ma'am,
Yeah.
we will get all the reading assignments in the problem solving session in the NPTEL portal, right? After the class is over.
It is there in the video itself, sir is mentioning what are the reading assignment papers.
Okay. No, actually, I am asking for your these problem solving session class recording and side by side these reading assignment papers. Where we'll get it in NPTEL portal, right? Problem solving
Yeah.
session.
problem solving and paper discussion, both will be uploaded on the YouTube video.
Okay. Okay, I'm going through the paper.
Okay. My screen is visible to all, and the slide is in the paper reading assignment slide, right?
Ma'am, please wait for 2 minutes.
Okay, okay. You're able to see the reading assignment slide now. Okay, let's get started with this paper. So in this paper, they have grown stem cells on different substrate stiffness, and they are seeing how they are getting differentiated in soft versus stiff substrate. So they are using a collagen gel, and with collagen gel, depending on how much concentration you're adding, the amounts of cross-linking will be different. And when the cross-linking is different, the substrate stiffness will also be different. So they are growing in three categories. The first category is zero point 0.1 to 1 kilopascal, which is about the stiffness of brain. And the second category is 8 to 17 kilopascal, which is the stiffness of muscle. And the third category is 25 to 40 kilopascal, which is the stiffness of bone. So in all of these substrates, just if you see the visually in the phase contrast image itself, you can see that the cell morphology itself is different. In the softer substrate, we can see a more branching pattern, especially after 4 days or 96 hours, there are more number of branches. In the second category, 8 to 17 kilopascal, and within 4 days, the cell is becoming more spindle shaped. And in the third one, the cell is getting spread out more. So if you see the first one, 1 kilopascals, that is the branching that you see is similar to the branching that we see in neuron cells. And the spindle shaped in 8 kilopascal, that spindle shape is similar to what we see in the muscle cells. And the spread out cell pattern is what we see in the bone cells. So we are seeing that depending on the substrate stiffness, the cells are able to differentiate into different types. But still, we cannot be sure because it is just the cell morphology. We have to look at the other gene expression profile and um, cell marker for the different cell types as well. So that is the next experiment they did. So for the softer substrates, to make sure that what they are seeing is that the cell has differentiated into a neuron cell, they have used beta 3 tubulin and NFH. So these factors are specific to the neuron cell type. So only if the stem cell has differentiated from stem cell to neuron cell type, only then we can see their expression. And in the softer gel, we are able to see that in the branches, the beta 3 tubulin and NFH expression is high. And here in this plot also, we can see that as it is going from soft to stiff, the area is decreasing, and the number of for the stem cells, the number of branches is increasing. So it is decreasing with stiff substrate. So it is showing that in the softer substrate, the stem cells are becoming more neuronal like. That is why we see a decrease in the number of branches. But in fibroblasts, we are not seeing any trend. In soft and stiff substrate, we are not seeing any change in the number of branches because the fibroblasts are already differentiated cells. And they have also done blotting and fluorescence intensity staining, and we can see that in the softer substrate, the beta 3 tubulin expression is the highest, and in the glass, because it is very stiff, it is in the range of gigapascal. The expression of the neuron cell type has become very low. And similarly, they did this in the stiff substrate as well. In the stiff substrate, it is going from the stem cell is differentiating into an osteoblast cell. So just in order to confirm that, they have used a protein marker called osteocalcin, which is present only in the bone cells, and they are able to see that in 34 kilopascal, the cells are expressing osteocalcin, which means that the stem cells have differentiated into the bone cells. And one more confirmatory test is the matrix, because the bone cells are able to secrete their own matrix, and they are able to see that the they are able the matrix thickness is increasing because the cells have they have differentiated into bone cells, and they have secreted their own matrix, as well as the elasticity also is matching with the matrix of the bone cells. So now they have more or less confirmed that in the soft cells we see a more neuronal type differentiation, but in the stiff gels we see a more bone cell differentiation. And this is the same thing, but here these are immunostaining images where in the soft gels we can see the tubulin marker, but in the medium and stiff gel we are not able to see it. And in the medium stiffness gel only we are able to see MyoD, which is a muscle cell marker. And the osteogenic, the bone cell marker, CBF alpha 1, that we are able to see only in the high stiffness, which is showing that depending on the stiffness of the substrate, the stem cell can become either a nerve cell or muscle cell or a bone cell. And then they wanted to see what about the media conditions, because the stem cell differentiation is not just depending on the stiffness of the substrate, but it is depending on the media that we are adding as well. So the first one, figure A, is for the muscle cell differentiation. We see that in the medium range stiffness, like around 10 kilopascal, the MyoD or the muscle cell marker expression is higher. But in the second case, where we are doing it with MIM is the muscle cell, that media. So it has the factors necessary for the cell to differentiate into muscle cell.
Where-
Yeah.
Yes, your screen is not visible. It's loading.
Okay, I will go to. Now it is visible. I have gone out from the slideshow mode.
Yes, yes. Actually, your problem is problem in your slideshow mode, right?
Yeah, I think so. Okay, so I'll just keep it minimized. Yeah, so in this experiment, they want to see whether the media conditions is also playing a role. So first they are just culturing on different stiffness. That is the lowermost curve. And in the uppermost curve, they are culturing in different stiffness as well as adding this MIM, which is muscle induction media. So if you compare the two curves, when you have added the induction media, the muscle cell marker is going up. So meaning that both of them are having an additive effect compared to just the stiffness. If you have the stiffness as well as the media, then the cells are becoming more and more muscle-like. So that is what the A is. This A figure is telling. And similarly, B is also the same one, but instead of for the muscle cell, they have done it for the osteoblast cell. And here we can see that the peak is in the stiffer range, around 50 kilopascal. But here for the muscles, the peak is around 10 kilopascal because that is the stiffness in which the cells are becoming muscle cells. And these are with the fluorescent intensities or immunostaining. And the same thing they have confirmed with Western blotting as well. So you have to compare this one, the second and the third one. In the second one, muscle cell, if you see the 11 kilopascal band is thicker compared to the third one. And the 34 kilopascal also, in the third one, there is no band. But in the second one, there is band because in the second one, it is not just the stiffness, but the media as well. So from this experiment, we are able to say that both the substrate stiffness as well as the media conditions are dictating what is the stem cell fate. And then the authors are hypothesizing that this how the stem cell fate is determined is by the stress fiber formation, and they are able to see that in the as the stiffness is increasing, the stress fiber formation is also increasing more. In the glass, you can see more straight lines. So these are the stress fibers. And here in the first experiment itself, they have did an experiment with myosin, where when you add myosin and decrease the stress fiber and actomyosin contractility, then we are not seeing any change in the, like here they have done a blebistatin experiment. So when we add this blebistatin to inhibit the actomyosin contractility, then the stem cell fate is not changing. So they are here in this paper, they are saying that in different substrate stiffness, the actomyosin contractility is different. And because of the different levels of contractility, the stem cell is deciding whether it has to become a nerve cell or a muscle cell or a bone cell. So that is the main idea of this paper.
So in case of the contractility, the stem cell will be different, nerve cell, muscle cell, or any other, right?
Yeah, if you inhibit the contractility, then even if you change the stiffness, it will not differentiate properly. That contractility is needed for the stem cell to differentiate into the correct cell type. Any other doubts?
No, no doubt for today.
Okay, then we can close today's session. Let's meet again next week, Monday.
Okay,
Thank
ma'am. Thank you, ma'am.